uno de nuestros mejores exalumnos, el señor José Felipe Rivas, y recibió también a nuestra colega y profesora Victoria Casanga en su pasantía doctoral en su laboratorio. El profesor y su equipo han estado en primera línea enfrentando brotes de enfermedades infecciosas emergentes tales como el virus de Schmallenberg, la lengua azul, la peste porcina africana y por supuesto el tan conocido COVID-19, entre otras. Son muchas las lecciones belgas en el manejo de enfermedades emergentes. Tienen una historia extensa de la cual podemos aprender muchas lecciones, pero no solo a nivel técnico y en investigación, sino que en método científico, en la prolijidad y responsabilidad de sus tareas, en la interdisciplina, en la cooperación y el trabajo en equipo, donde también tenemos algo que decir, ¿verdad? Los dejo entonces con el profesor Dr. Mutien Garigliany. La charla es en inglés, amigo, y las preguntas pueden ser formuladas lentamente en español o haré el intento yo de traducir aquí. Adelante, profesor.
Muchas gracias. So yeah, the presentation will be in English. Thank you, Professor Krasnova, for the presentation. So I decided to present you two things tonight. First, our experience with the COVID-19, so the SARS-CoV-2. I don't know if the sound is fine with you? Yeah? Okay, great. And next, just I picked up one of our main projects that we are which is currently ongoing in our laboratory. So you see a bit the kind of experiment we are doing. So about the COVID journey of the University of Liège, the funny part is that most of the work has been done by vets. And this is kind of logical because vets, we are used to face emerging infectious diseases. So this is something we see every couple of years. We have a new disease emerging in a given animal species. And we know how to manage these emergence. We know how to diagnose this infectious disease, to isolate these agents, and to develop quickly models, so to develop a quick answer to this emergence. And this is something that probably has been lost over time in human medicine. And what I really appreciated here is that very soon after the emergence of SARS-CoV-2, we as vets were invited by our medical doctors, colleagues, to participate to the fight against COVID. And that was really a nice story to me and a great opportunity for us, I would say, little veterinarians, to be part of that effort against the SARS-CoV-2. So this is very simply to show you what we did and how sometimes very simple, classical, really old techniques of biology, for instance, are still very useful nowadays. So just a bit of geography, just to show you how close is the veterinary faculty here from the human hospital, just 500 meters. But it's quite good because there is a proximity between both, and this is the basis of strong partnership. Yet, so far, and in spite of different attempts, okay, there were some key collaborations between colleagues from both faculties, but there was still a strong barrier here between human medicine, high above, and the little vets here down below. And one of the good sides, I would say, of COVID-19 is to destroy all these barriers. And that was probably something good. This is our faculty from the slides. Looks nice, and it's in the middle of the forest, so it's very green. Of course, we have a nice hospital, and we do plenty of things. We do in many faculties, vet faculties in the world. We have some top-level equipment, but this is not what made the difference here. What made the difference in what we were able to do during this pandemic is this very little laboratory, 3 rooms, very small, the BSL-3 laboratory, that we developed at the time of at the time of the blue tongue emergence. You know that blue tongue virus has to be handled under biosafety level 3 conditions. And then at that time we decided to dedicate part of our space to a new small BSL-3 laboratory with a main laboratory for L3 conditions, so in vitro work, and a very small animal husbandry here in this L3. And this is the people working there, of course. Okay, so this is not much in terms of space. This is not heavy equipment. This is not top technological equipment, but yet it proved extremely useful during this pandemic. So this is the menu for this presentation. So the context, you know it, of course. This is really funny because we work a lot on infectious disease, and this is part of a big project that we had studying the infectious agents that were present in bushmeat samples. You know that bushmeat is something very important in the economy of many countries. And there are also cultural reasons why people, for instance, in Africa, still eat a lot of bushmeat, even for traditional moments, like weddings, for instance. They like to have this special bushmeat. This is pangolin. You recognize it, of course. And this is, you can see here, it was a European Commission meeting, and it was early December 2019. And with colleagues, we were trying to convince people from the European Commission that we should take care with this wildlife trade, and in particular bushmeat, because there is a high risk of emergence of a new pathogen in human medicine because of that, especially through transportation by airplane over continents. And you know that a few days later, COVID-19 was reported from China, and from Bangladesh. So this is funny. So the question, this is what happened historically, is that a new human respiratory disease has been reported from China. They called it an atypical pneumonia in people from the Wuhan region and this market that you know, you all know the story. And all PCRs targeting the known pathogens, known human pathogens for respiratory disease proved negative. What would you do? How could we diagnose, identify the infectious agents here? So you know that we can use what we call de novo methods. So when you don't have a clue about what can be behind it, we use this de novo method, so you don't have an a priori regarding the infectious agent. And there are two types of methods, metagenetics and metagenomics. Probably you know the difference already, but just a quick reminder. Metagenetics is like the 16S RNA sequencing for bacteria. Actually, we just use primers to amplify a very specific region of the genome, in this case of bacteria. And since this very specific genome piece is very specific of each bacterial genus, and to a lesser extent bacterial species, you can determine which are the bacterial species and genera present in the sample. And since you can count the number of reads which match with a given bacterium, you can also have a clue about the abundance and the relative abundance of these bacteria. So this is metagenetics. Metagenomics, and that's how they discovered the SARS-CoV-2, is just basically taking the whole content in DNA and RNA of the sample, you cut it into small pieces, and you sequence everything. Okay, it's also called shotgun sequencing. And this is the kind of result you get. So this is the metagenomic picture of a sample with bacteria, viruses, and the host here, DNA and RNA. In this case, they found plenty of reads corresponding to a virus, and then you can build back, by assembling these reads, the full genome of the virus. So you know that at the end of December 2019, so a few days after our meeting with the European Commission there, the WHO was informed of this case of atypical pneumonia, and they found out this new virus. It was very closely related, as you know, from a bat coronavirus, and very closely related to SARS-CoV-1. So this is the context, and you all know it. Our first experience, I would say, with that virus was through an unusual case. In February 2020, a Belgian citizen was part of a one-week trip in Italy in a ski resort. There was, I would say, an advice from the authorities not leaving the territory and not traveling through Europe, but there was nothing mandatory. So some people decided to go anyway, and this is what this person did. And the day after her return, she started to feel bad and short of breath, so she called her family doctor, which took a swab. So it was on March 2, if I remember correctly, and which eventually proved positive for SARS-CoV-2, was one of the first confirmed cases in Belgium at that time, on March 6th. It required 4 days before getting the diagnosis. We will talk about it a bit later. It's very long. She was in a stable state. She was relatively fine, a flu-like symptom, and stayed home and under surveillance by the doctor. And that time, the doctor decided to visit her every day, okay, to see how she evolved. It was one of his first cases of this infection. Interestingly, and this is the unusual case, the patient's cat started to develop clinical signs on March 9, so a couple of days later, with prostration, anorexia, and vomition, then diarrhea, and a bit later started to develop respiratory signs with dyspnea, sneezing, harsh, productive cough. I have a video. I hope it will be possible to show you this with the sound. Hopefully. You can hear the productive cough. Okay. So you don't need to be a vet to see that this cat is sick, definitely. So the doctor is also part of the staff of our hospital, the human hospital near our faculty. And he called us and said, okay, here I have a cat with symptoms, and this is very weird because the owner, she got sick of SARS-CoV-2 a couple of days before. What can we do? Oh, yes, take swabs. Take samples on the cat and whatever. So we could not access to the cat, unfortunately, so we cannot ourselves do the sampling and checking the evolution of the cat. But the doctor did everything we asked him to do. It was very, very funny, very interesting. So I'll get to the next slide. I think I have another video yet. Yeah, this was the evolution of that cat. Very similar. We start, I will show you after when it started and lasted a few days and quickly improved after that. At some time we thought that the cat would die of the infection, but it improved over time. Okay. I think I will remove that guy. Okay, this is the evolution of the owner, starting March 2, and here positive by PCR and showing different signs, not too severe. It was a mild case. And here's the evolution of the cat starting March 9, and the doctor was really kind because he took a lot of samples on the cat, and we could see here the evolution of the PCR, which proved positive for about 10 days. Which is extremely interesting, and this is the evolution of the clinical signs, so mostly general clinical signs, digestive signs with vomition, diarrhea, and respiratory signs here. And this is where the different videos were taken. Everything is available on the web if you like. And this was very interesting because there was here a present of quite high amount of viral RNA for about 10 days. Which proved that it was important at that time to identify that case, not only on a veterinary point of view, but also because let's imagine that that person would have been more sick of the infection and should be hospitalized, then some other people should take care of the cat. It means that when you take care of the cat of a person infected by SARS-CoV-2, you should wear a mask, because you could get infected by the cat. That was important on the public health point of view. That's why we quickly published that case, and we actually were the first to identify the reverse zoonotic transmission of SARS-CoV-2 to an animal, and in this case, to a cat. It was really, really interesting. Second point is the PCR detection. I showed you that this person required 4 days before getting her PCR result. It was really, really long. And at that time, we realized that there was a poor organization of the medical laboratories, the human diagnostic laboratories. They were not used to manage large numbers of samples. And this is really contradictory to what we are used to do in veterinary medicine. We are able to sequence and to detect by PCR, millions and millions of samples for a year. This is something very usual in especially large animals outbreak, but this is not something that are used to do in human medicine. So that was the first problem. At first, for the whole country, for the whole Belgium, they said, okay, we can do 4,000 or 5,000 PCRs per week, which is unbelievable, because this is what we can do usually in my own laboratory, so which is a small laboratory. You might remember also that there was a shortage of extraction kits, because there was a high demand globally, chiefly magnetic beads, and to a lesser extent PCR reagents, and also very quick plastic consumables could not be found on the market. That was a big problem to build up the capacities in terms of PCR detection of the virus. And another problem was that all samples, the swabs taken on patients, had to be inactivated in a BSL-3 facility before being transferred to a PCR laboratory, being opened, and then that we could proceed with the extraction. That was a real bottleneck in the management of such large number of samples. That's how we had to proceed at first, using this BSL-3 laboratory and treating all samples there before they could be sent to the PCR laboratory. It was really painful and difficult. In February 2020, as we say, that there were the first cases in Europe, mostly in Italy. Then in March 2020, first cases in Liège, and including in the hospital. So they created the Liège COVID team, so mostly medical doctors, of course, but they invited us as veterinary to give a piece of advice and maybe to help if we could. That was really, really nice of them. So the doctors from the human hospital asked, okay, we really badly need here some people able to isolate the virus and quickly develop model. And we said, okay, we can do that. And we have the BSL-3, we have everything. So very quickly, at the end of March 2020, we had the virus in cell culture. Here's a positive staining for the nucleoprotein of the virus, for instance. The first thing we did was to facilitate the process of the sample by validating an easy and cheap method to inactivate the virus through temperature. This is what we developed a couple of years ago for the African swine fever. Just a heating at 70 degrees for 15 minutes kills completely the virus. And you can see that it has no effect on the CT value, so on the sensitivity of the PCR. So when you receive a tube containing the swab of the patient, just put it at a temperature for 15 minutes, and then you can open it without any risk of infection. That was way easier than treating all the samples with inactivation buffer in the BSL-3. That was easy, straightforward. That was done in March 2010. Then colleagues of us started the production in-house of inactivation extraction buffers that were, because of the shortage on the market, based on gonadinium azothiocyanate. We established also inactivation protocols for different buffers that were produced by private companies that required validation, and also standard operating procedures for the PCRs. We were recruiting people, there were plenty of volunteers helping, so we had to establish standard procedures so that anybody could help in the process without taking any risk for their health. And also we produced virus stocks and as controls. During the first months, we were the main producers of the viral controls for the whole Belgium, which was really, really nice. This is our vice rector of research at the time, which is a vet. And other colleagues from the Department of Chemistry started producing their own magnetic beads. You know that magnetic beads coated with silica can be used for the extraction with robots. And since they could not be found in sufficient amounts on the market, the university produced its own reagent. That was really a major step forward to build up the capacities. So they produced these large scale. and we set up a dedicated diagnostic platform directly in the Faculty of Veterinary Medicine. And in the end, actually, this Center for PCR for SARS-CoV-2 handled the largest number of samples in Belgium, the whole Belgium. Much more than all the medical, the human medicine laboratories that also perform PCRs in Belgium. So it was really a major hotspot of PCR detection of SARS-CoV-2 during the whole crisis. This is our king, King Philip from Belgium, visiting our institution and the laboratory with our Vice Dean for Research, Laurent Gilles. They also produce in the university directly the plastic consumable. Okay? So actually, basically, we produce everything on site. So we're not really dependent on the shortage that was on the market, which allowed us to rapidly scale up the PCRs. This was also an interesting step, is that when we started to work with the different laboratories in Belgium, we had to have common standards in terms of sensitivity and specificity. And the problem is that all laboratories did not use the same machines, including the same PCR machines. So we had to normalize everything. I will not go through the details, but some private companies produce very interesting LIMS. So these are laboratory information management systems that corrected for the difference in performance between the different machines to provide common results with a same level of sensitivity and specificity directly to the patient, and to improve the process over time. It was really measured. And this is something very interesting I wanted to share with you. This is something we use a lot in veterinary medicine since many years to improve the efficiency of PCR detection. It's called smart pooling of samples. I don't know if you know that. So when you proceed to PCR detection of a pathogen, there are two ways to proceed. Either you receive the sample and test each sample individually. Okay, this is long, time-consuming, and expensive. Okay, if the prevalence of a pathogen is low in the population, you can pool the samples. Okay, because the likelihood that a given pool is positive by PCR is low, so you can decrease easily the number of PCRs you have to perform. This is an easy way to earn money and time. In that case, the prevalence was high. So how can we do that? This is called smart pooling. How does it work? Actually, if you take a sample from a patient and that you include that sample in several pools, by checking which pools are positive, you can retrieve back which were the positive samples. Okay? And they can do that in a way that, in the end, you just need 3 PCRs to test 10 samples. Okay, so this is a really improvement in terms of scalability of the PCR, and in terms of cost also, efficiency. This is really, really nice. There are plenty of papers on that since the COVID-19 experience. This is something we did already in the past for the management of veterinary disease, but it has been well implemented and improved, I would say, with SARS-CoV-2, and this is very interesting if you have to work in that kind of conditions. Okay, so there are plenty of papers on that. SNT analysis. SNT is for seroneutralization. I will explain a bit what it means. So in March 2020, you know how desperate were the medical clinicians who were facing patients arriving at the hospital. They were trying everything they could to save these people, and these people, most of them died of the infection. It was really a nightmare. And so we were having weekly meetings with them. It was really, really hard. We are not used to that in veterinary medicine. It's different to hear about the death of people. And so they said, okay, we don't have any efficient antiviral. We don't have any way to treat and support efficiently these persons. So one way to do, because some people are recovering from the infection and probably build up a strong immunity against the virus, is to take the plasma, the serum, from recovering patient, which is rich in neutralizing antibody, and to use this plasma to protect incoming patient at the hospital. But we need a way to quantify the neutralizing activity in order to select the best plasma to administer it to incoming patients. So again, the same question, how would you do that? And the answer is cell internalization assays. This is very old methods. All virologists know that. The principle is very simple. If the plasma or the serum contains neutralizing antibodies, these antibodies will coat the virus and impede the virus to infect the cells. So in this case, for instance, you can see the cells because these cells have not been infected. They have been protected by the neutralizing activity of the plasma. In this case, on the contrary, the virus was able to infect the cells and cause cytopathic effect. Okay? If you perform different dilutions of the plasma or serum, you can see up to which dilution this plasma is able to protect the cells from infection. So this is called the neutralizing titer. And of course, the higher the dilution, the higher the titer, and the better it is for therapeutic purpose. Okay? That's what we did basically. So it's very simple. You just need a multi-channel pipette, 96 well plates, and a lot of time, and a lot of people to do that. So there were plenty of volunteers helping us to do that. So it's not high technology, but it was very interesting. And the Red Cross, the Belgian Red Cross, also asked us to do that. They started to sample at large scale donors that were recovering from the COVID-19 infection and that wanted to give their plasma to save people. And we did also some big clinical trials at the Belgian and European level, and also a surveillance program in the university to see how the epidemics evolved over time. So, is it efficient? Well, it depends. It is efficient provided it is administered very quickly upon admission of the patient, within 3 days of admission to the hospital, and if only high titer of plasma is used. Okay? And in this case, you can significantly decrease the mortality of the patients. So we did plenty of such analysis, and the first thing we did actually was to make a ring test between the different laboratories performing such tests in Europe to harmonize the way we were assessing the neutralizing efficiency of the plasma. And once this had been done, we could start with different clinical trials. I will not go into the details, but this is a big one that just ended end of 2022. I will talk about it a bit later. Another one here. Again, another one. And this is the one I wanted to talk to you about, the confidence. So this is the one we performed in the whole human hospital. These are the different waves of the different variants of SARS-CoV-2 that you know. The alpha, beta, gamma, delta, omicron variants. This is the collection of plasma from recovering patients. This is the different hospitals that from which the donors came. And this is the inclusion of the patients that were receiving plasma therapy upon admission. And this is the comparison between the patients receiving convalescent plasma versus the patient receiving the standard of care. So this is the classical way to treat these patients. And you can see that the numbers are really similar and very similar populations. And what did we observe at the end is that for those patients receiving convalescent plasma very early, you can see here the mortality rate, broadly 33% versus 47%. So you can significantly decrease the overall mortality, and if you compare those numbers, it's a decrease by about 30%, which is quite high actually. It's very good. So this is probably the part of the story of which I'm the most proud, is that hospital vets helped to save some people. And this is wonderful. And using very basic techniques, cheap basic techniques, but a lot of work. There was a big study within the university to follow up the different waves of the epidemic and also the impact of the vaccination. So I will not go into the details here, but this is very, very interesting if you like such kind of thing. And this was based on autotests based on saliva, not to do a swab all the time. We also had to study the vaccine efficiency because you know that the aim of the vaccine that we used, mostly the RNA-based vaccines, based on the production of the spike protein of the virus, is to induce the production of neutralizing antibodies against this spike protein. And this is something that we assess, especially in the risk population of elderly people, because we know that elderly people over a certain age have a tendency here in green to produce very low titers in terms of neutralization antibodies compared to younger patients. So this is a big problem, and so we had to develop, well, the pharmaceutical companies had to develop strategies to improve that. Okay, that's why Moderna, for instance, had to use more RNA in their preparation to try to boost that response. Again, all of this has been done using very basic techniques. Okay, the last thing that we did is those inactivation tests and antiviral studies. So quickly, in April 2020, we developed in vivo models based mostly on the serum hamster, because you know that the serum hamster is spontaneously susceptible to the infection. Here is a very nice image from the nasal cavities, so the nasal turbinates of these hamsters with the olfactory epithelium. And you can see how the virus targets the olfactory epithelium, destroying this epithelium. And this is here the exudate you find in the airways. And it's easy to understand how you lose the smell quickly after infection when you see that. And this is the lesions we observed in the lungs, so which is called acute respiratory distress syndrome with diffuse alveolar damage and pulmonary edema, which is very close to what happens to people. This was a very interesting model to test the impact of therapies, not only therapies aiming at reducing the viral replication, so antiviral, like remdesivir, for instance, was validated on hamsters, but also therapies aiming at decreasing the level of inflammation in the lung. And that's why steroids, for instance, dexamethasone, proved to be quite efficient in reducing mortality in the animal model and then in people. We quickly validated most models. It was a bit longer because mice do not express a similar molecule as ACE2, so the main receptor of the virus, as that of human. We also were able not only to show the presence of similar lesions in the lungs of hamsters as in people, but also to show a similar cytokinic storm in these infected animals. For the screening of antivirals, we use, again, very simple techniques. There are other institutes, like the KU Leuven, for instance, in Belgium, they use machines allowing the testing of hundreds of thousands of molecules at the same time. But here in our faculty, we still use classical methods. The principle is very simple. You seed cells into the wells, add the compounds at different dilutions, and then you add the virus and you see up to which dilution of the molecule it is able to inhibit the replication of the virus, and in comparison with cytotoxicity assay, of course. Very simple and still widely used currently. So we have plenty of projects currently ongoing based on that, and plenty of publications, and I think that's it for that presentation. Are there any questions? Sorry, it was a bit fast, but we still have a lot of things to say. So. We can, yeah?
So, if the virus has this tendency to attack the nasal epithelium, does this mean that if, for example, a cat gets infected with the virus, is it possible that the cat loses his sense of smell?
Likely. So we do not know exactly what happens in the cat because most have survived the infection first, and there was no detailed study on the spread of the virus within the different cat tissues. What we know is that it's a matter of time. So in the early waves of the infection, indeed, the virus has a tropism for nasal cavities, probably because that's the place where the receptor is the most expressed. What we know is that in a given host, provided the infection lasts sufficiently, the tropism of the virus evolves over time because there are mutations accumulating in the viral population, and the virus gains a tropism for deeper airways and then the lungs. So for those patients that are able to fight efficiently the virus from the start, it remains in the nasal cavities. For those that are not so efficient in controlling the viral replication, the virus evolves and gets deeper in the airways up to creating so many lesions in the lung that the person may die from the infection. And this is also what happened over time. The variants that were present in the population were not the same as the beginning. And this symptom of the loss smell was not so characteristic anymore as it was at the first, with the first variant. Okay. Okay, I suggest that we go on with the next presentation. So the first presentation was really to show you that, well, vets can do things and that you don't need very high technology methods to be able to do something. That was the point. So the second thing I wanted to show you is what we did with an emerging flavivirus, with a word name that is called Uzutu virus, but I will tell you about that. And what a few PhD students did in that. So it's one of my main research projects in the laboratory. We have a couple of them, but this is something I think that can be interesting for you. So what is Uzutu? Well, Uzutu is a river in what was previously known as Swaziland, near South Africa, and this is where this virus had first been described. And it has been well known in Europe because in 2001, in Austria, we could observe such kind of things along the road, so hundreds of thousands of blackbirds dead. Why these birds died through close to these roads? Well, because they had neurological deficits, they were not able to fly as efficiently, and they were killed by contact with cars. Okay? That's why we easily identified these animals, and quickly the virus was isolated, detected, and it had a significant impact on the blackbird population. Not only the blackbirds, many passerifers have seen their population really drop after the emergence of this new virus. Interestingly, they did some retrospective analysis in samples from Italy, and they showed that the virus was already present in 1996 in Italy, with also mass mortalities of birds. Okay? It appeared in Belgium in 2012, then it disappeared and re-emerged in Belgium in 2016, and now it has become endemic. Okay, so we detect it every year in a couple of birds. So this bird is transmitted by mosquitoes, and there is a reservoir in bird species. So the main cycle involves the vector, mosquitoes, and the birds, and sometimes infection of mammals, including people and a different bunch of other mammals. And this is highly reminiscent of what we observe with West Nile virus, which is very closely related to Uzutu. And some sporadic cases in people. So it's not so frequent, but some people get infected and can get sick of the infection with neurological symptoms, not only in immunocompromised persons, but also in persons which are healthy and start developing these symptoms after infection. So very similar for Uzutu and West Nile. West Nile is more pathogenic than Uzutu virus. We'll talk about it. But there were cases of fatal infection with Uzutu virus, like this one recently published in May 2023. It's a case from last year in Italy, where one person unfortunately died from the infection. So it's a significant pathogen in birds, and it can be significant in terms of public health as well. How did it occur to emerge in Belgium and in Europe in general? Well, it was transported through migratory birds. You know that there are many viruses that can be transmitted over continents by migratory birds, and you know that this virus first was detected here around 1950, something like that, and then started to be present in Western Africa, and then to gain access to Western Europe, and through the Danube Delta here in Eastern Europe. This is very interesting because we did a large-scale phylogeographic analysis of the virus, and saw that there were African strains of the virus which emerged in Europe, and then there was a new diversity of the virus. There's kind of a bottleneck here because the virus get adapted here to local bird species and local mosquito species, which are not the same, of course, as the African strains. Just as a matter of curiosity, I wanted to check the flyways that might be important for Chile, and this is the Pacific Americas flyway that you should probably survey over time because it can be significant. You probably know that this migratory bird transport other viruses, significant viruses like highly pathogenic H5N1 influenza, you know, and there was a case, a human case of H5N1 infection in March here in Chile, probably coming through this flyway. Okay, so very, very significant. Okay, this was just. What is interesting about this virus is that there are two kinds of birds. Those highly susceptible, like passeriforms, like the blackbird and night raptors, and those highly resistant, like chicken, for instance, and ducks and goose, which are very resistant to infection. There are very few examples of cases of mortality in Anseriformes, so ducks and related animals, including this one, but this is very limited. This is the different strains that we isolated from Belgium, so mostly from Africa 3 lineage, Europe 1 lineage, and Europe 3 lineage. And you can see here that this Usutu virus is very closely related to a significant pathogen called Japanese encephalitis, closely related to West Nile virus, closely related to Zika and dengue virus, and yellow fever. So all very significant pathogens, either in animal species like West Nile, and human, of course, or pure human pathogens like Zika or dengue, for instance. The main interest of Usutu is that among all these viruses, this is the only one that can be handled under biosafety level 2 conditions. All the others must be handled in BSL-3 conditions. And you have seen that our BSL-3 is quite busy with SARS-CoV-2. So we were quite happy to have this virus. So we can study the biology of all the family, let's say, of mosquito-borne flyviruses, also the families, different serogroups of mosquito-borne flyviruses without the need of the BSL-3, and with less risk for the human health. We also study West Nile and dengue, but in different projects. It's becoming highly significant. This is the image of the spread of the cases of West Nile infection here in Italy. So this is the Po River Delta near Venice. Here is the Danube Delta. Okay, wherever there are many migratory birds, there are these viruses. Okay, and there is a northward spread here. You see that there are no West Nile cases in the Netherlands also. So just a few numbers. More than 1,000 cases of diagnosed West Nile infection just for Italy last year, and this is likely largely underdiagnosed because most cases are not detected, of which 37 died. Again, only those that were diagnosed. So probably the true numbers are way higher. And 2023 is expected to be worse. There is no vaccine, and there is a high risk of what we call the antibody-dependent enhancement. I don't know if you know what it is. This is very specific of flaviviruses, can be encountered with other viruses. It has been shown for SARS-CoV-2 as well, but this is very classical for flaviviruses. And what is it? You know that I explained to you that usually when neutralizing antibodies are produced by the host infected, they will be able to stop the infection of new cells by the virus. They have a neutralizing capacity. But if these antibodies are not neutralizing, they can be used by the virus to enter cells that are not normally part of their tropism. Why? Because you know that some cells have receptors for the Fc fragment of antibodies, like macrophages, for instance. And so if these antibodies are not neutralizing, they can help the virus to enter these macrophages and to induce a much more severe clinical condition than in the classical disease that we observe with the virus. And this is what caused the hemorrhagic disease with dengue virus, for instance, or West Nile virus, for instance. So there was a fear that Usutu, for instance, could sensitize the human population or animal population and make it more susceptible to develop an hemorrhagic disease after West Nile infection. especially for these areas, like the north of Italy, where both viruses are highly present. And I would say that fortunately, several studies have shown that Usutu virus, when there is a pre-existing immunity against this virus, which is actually much more spread than West Nile, it seems to protect people against West Nile, which is good news. It's like a vaccine strains you will have for West Nile. This has been shown in mouse models as well. So when you immunize mice with Usutu virus or the Usutu E protein, which is the main attachment protein of the virus, it protects mice against West Nile, including lineage 1, which is the most virulent lineage of West Nile virus. This is very, very interesting. So the questions were, what are the determinants of the virulence in humans and mammals? So why do only a few people and mammals develop a disease? Why there are so many seropositive persons and animals? This is very weird. So we don't know why. And the second question we wanted to answer is, what are the viral and host factors which are involved in the high differences in susceptibility and resistance in bird species, because they are highly resistant species and highly susceptible species. So why is that? So first, about the mammals. So one of the previous PhD students of the lab, Emna, had shown that immunocompetent mice are clinically highly resistant to Usutu virus infection, that mice which are knockout for the receptors to type 1 interference are highly susceptible, but we observed a sporadic occurrence of fatal disease with neuroinvasion in adult immunocompetent mice, which is highly reminiscent to what is observed in people. Most of the case, it's asymptomatic or mild clinical symptoms, and sometimes you observe neuroinvasion and a very severe disease, as you may observe here, with positive neurons, degeneration of the neurons, satellitosis, and perivascular curves of inflammatory, so typical of encephalitis and myelitis. The first thing we wanted to assess is, are there differences between the different laboratory mice? You know, there are plenty of genetic backgrounds of laboratory mice, and we know that depending on this genetic background, these mice might be more or less susceptible to the infection. That could be a good way to have a more severe model of infection of Usutu infection. We know that for Zika, for instance, Zika virus is the case. Different laboratory mice show different levels of susceptibility. We know that for pneumonia virus of mice. We know that for Ebola, for instance. And our colleague, Professor Casanova, has shown that for influenza virus as well. So we tested that hypothesis, comparing the susceptibility of different mouse strains. And you can see here that the seroconversion rate is highly dependent on the mouse strain, with some highly susceptible strains and more resistant strains. But in all these cases, there was no clinical sign, no mortality. So this is probably not a good way to have a model, a lethal model of infection. The second question was, what is the impact of the age of mice? Because we know that Zika virus, targets neonates and infants in utero. We know that dengue virus is more severe in children less than 15 years, and the same for Japanese encephalitis, more severe disease in children. So what about Usutu virus? We don't know. Okay. That's why we set up this experiment. We infected different groups, age groups, I would say, of mice, and we inoculated the virus, and we saw that there was an age-dependent development of clinical signs and mortality in these animals, and very linear evolution of the mortality between 5 days old to 15 days old, so a very narrow window of age. So this was using subcutaneous injection into the skin of the back of the mice, with the development of typical clinical signs of neuroinvasion with paraparesis and paraplegia, proprioception deficits, ataxia, tremors, and seizures. in mammals, there is a clear neurotropism. So there is a clear high replication of the virus in the central nervous system compared to other organs like the spleen or the liver. And you will see that it's completely different from what we observe in birds. So here, pure neurotropic disease, very similar to West Nile infection. We observe the same lesions here with inflammation, destruction, so degeneration and destruction of neurons and satellitosis. With very beautiful immunostaining of the virus here in the central nervous system. We also validated the foot pad injection instead of the injection into the skin of the back because it's much more convenient for sampling. And you can see that the age is a bit different because here even 9-day-old mice all died from the infection. And here you can see that 15-day-old mice all survived the infection. So the window is even narrower using that route. So it's a very good model, and it's highly repeatable and easy to handle model. Very interesting. So the question is, what is happening between there and there to make mice so quickly resistant to the inoculation? So we inoculated two groups of mice, 9-day-old mice and 15-day-old mice, and we followed up the spread, the dissemination of the virus in different tissues to see what was happening. And you can see that at the stage, at the level of the skin inoculation, the foot pad, there is no major difference in terms of replication of the virus. So this is not where the difference between both phenotypes appears. You can see a mild difference in the blood, okay, a bit more in the young mice compared to the slightly older mice. Also a bit in the liver, but it's later in the evolution. But the major difference here is in the brain. Okay? So we do believe that the difference occurs in terms of neuroinvasion capacity of the virus and replication into the brain. Okay? That's why now we are studying in detail what is happening at that stage. And this is extremely interesting because we do believe that this might also be the explanation why some adult immunocompetent or immunocompromised person sometimes develop neuroinvasion and get sick and sometimes die from the infection because of this permeability of the blood-brain barrier. So this is the progressive spread of the virus from the foot pad inoculation through different organs, the spleen, the liver, the blood stream, and then gain access to the brain around day 6 or to 8 after infection. What we are using now to try to understand what's going on into the brain and how the virus gains access into the brain is called spatial transcriptomics. I don't know if you know that kind of technique, but I'm a pathologist, so I like histological lesions. It allows you to superimpose the lesions so you can see the tissue with the expression of different mRNAs. Okay? You can check the expression of any type of gene of interest. So it's like transcriptomic analysis, RNA sequencing, with an information about the spatial distribution of the cells the RNA is coming from. And this allows you to see where the virus is appearing, so where the infection starts, because we know that there are some preferential places where the virus might cross the blood-brain barrier, and we can see what's going on specifically at this stage, at this level, during the invasion of the central nervous system. So this is the PhD of Mazarin. What about birds? I told you some highly susceptible bird species, especially in the orders of Passeriformes and Strigiformes, and highly resistant species like chicken and goose and ducks. First part is the work of José Felipe Rivas. So the development and validation of characterization of an in vitro model of Osuto infection in susceptible and resistant bird species. René is laughing at our friend Felipito. So why in vitro? Well, for ethical reasons, it's not allowed in Europe to work with wild birds. We cannot catch wild birds and infect them in the laboratory. It's not allowed. So we have to find other ways to study the susceptibility of these birds, and one way is to use in vitro models. Okay. And then we'll try to understand the viral factors which are involved in crossing the host species barrier. Okay, the two parts of the PhD of Philippe. First thing, we know that when you inoculate the virus into the dermis, like a mosquito would do when it's inoculating the virus in a bird, there is a massive influx of leukocytes. And what is currently believed is that these leukocytes then get infected by the virus and will ensure the dissemination of the virus through different organs, and then the disease starts and the animal may die from the infection. So what's happening there? We know that one of the main targets of the virus are dermal fibroblasts. And these dermal fibroblasts are involved in both ways. First, we believe that they are a major amplificator of the virus when it's inoculated by mosquito. Then, we also believe that these fibroblasts, after they get infected, they will produce cytokines and chemokines that will activate the endothelium and the inflammatory response here, okay, and then increase the permeability of the blood vessels and allow the virus to escape into the bloodstream. But also the chemokines will be responsible for this influx of leukocytes. Okay? So one possible explanation to understand the difference between resistant and susceptible bird species is that there is no such influx of leukocytes in resistant species like chicken, okay? Because we know that there is no dissemination of the virus in chicken. So it's not easy to isolate dermal fibroblasts from dead birds, because these blackbirds, we find cadavers, okay? And it's extremely hard to isolate these cells. But there's another cell type which is interesting and which is very close to fibroblasts, and these are the mesenchymal stem cells. Okay? And what is highly interesting is that these mesenchymal stem cells, they are highly reminiscent of fibroblasts. They can do about the same things, but they can be isolated up to 3 days after the death of the animal, which is extremely convenient. That's why we decided to isolate tendon-derived mesenchymal stem cells. And this is how you can, from a piece of tendon, start to isolate these cells and then cultivate them. This is what we did for these blackbirds. Then we had to demonstrate that indeed we had isolated such mesenchymal stem cells through the expression of different markers. It looks easy like this, but there was no information about the sequence of these markers in birds, in blackbirds. So we had to do a lot of analysis, bioinformatic analysis, to try to build back the sequence of these genes in blackbirds and design primers. This was the work of Philippe. We also showed that these were indeed stem cells, mesenchymal stem cells, because we were able to artificially differentiate them into osteoblastic cells, into adipocytic cells, and into chondrocytes. Then we wanted to compare the permissivity of these cells to Oozo2 infection with the gold standard for the replication of flaviviruses, which are virucells. Okay. And you can see here by immunocytochemistry that these mesenchymal stem cells from blackbirds are permissive to the virus. They can be infected compared to the virucells. And you can see here the comparison of the replication curves. And you can see that, okay, the viral cells are slightly more permissive, but still you observe a significant replication of the virus in these mesenchymal stem cells. It has to be noted here that this virus that we used for that experiment is a stock that has been passaged a couple of times in viral cells. It was viro adapted, and probably it explains why it was more efficient in terms of replication in the viral cells. Okay, so it's a good model. And this was part of the first paper here of Philippe, which is currently under review. Then we compared the same cells coming from chicken with those we isolated from blackbird. And you can see here that there is a slightly higher permissivity of blackbird cells compared to chicken cells. We wondered if maybe this could be due to a difference in terms of receptors to the virus. This is frequently something which is key in terms of host species barrier, like you have seen for SARS-CoV-2. Those species that were susceptible to SARS-CoV-2 infection had an AC2 receptor in their airways similar to that of humans, though the other species were resistant to infection. Well, you can see here that the binding of the virus to the cells from the blackbirds or the chicken were similar. So it was not a matter of receptors. So the second question we wanted to answer is, okay, we have seen that there is a low but significant difference in terms of permissivity, of amplification of the viral population. But how do these fibroblasts from blackbirds and from chicken respond to the infection in terms of activation of the endothelium and recruitment of leukocytes? So Felipe performed RNA sequencing analysis comparing the expression of these cells in both avian species. And this is fine, this is very, very recent data. We observed a much higher expression of interleukin 8, especially the variant L2 and CCL4. These are two chemokines which attract monocytic cells, mostly dendritic cells, or monocyte-derived dendritic cells, which are known to be main targets of the virus, which allow strong amplification of the virus and allow also dissemination of the virus. So this is a key finding. We also observed that there is a much lower expression of antiviral proteins, including the oligodenylate synthetase, which is the main antiflavivirus effector of the interferon pathway, viperin, and other ones, including interferon omega, in blackbird cells. So this is very interesting. So higher influx of leukocytes, likely in blackbirds, and lower interferon response. We also observed a difference in terms of caspase 8 in blackbird cells with way higher expression, so a pro-apoptotic and anti-interferon response in these cells. Okay. So everything is done actually to facilitate the replication of the virus and then facilitate its dissemination into the bird. This is very interesting data. Second question we may ask, if taking the point of view of the virus, let's say we will try to adapt the virus to chicken and see how the virus evolves. This could also give us a clue about what is the key for the barrier for an efficient infection of chicken species or resistant avian species. We had previously with Felipe and Emna validated the infection of embryonated chicken eggs as a good model for replication of the virus. With widespread infection, dissemination of the virus, and you can see here easily the difference between uninfected chicks and these infected ones, with mortality, a dose-dependent mortality also of these embryonic chicken eggs. So what we did basically is to perform several passages of the virus, so two strains of the virus, of Uzo2 virus, from two different lineages in embryonated chicken eggs, until we observe a significant increase in terms of mortality of the embryos. That's what we did. And since we know the different proteins of the virus and their function, when we can identify key mutations associated with the increase in virulence in chicken, we can infer what is the key of the resistance of chicken. So we know that many non-structural proteins, for instance, are able to block, for different flaviviruses like dengue, Zika, or West Nile, the interferon pathway, either the production of interferon or the effect of interferon on the target cells. Another key feature that we know in terms of host tropism is the impact of the 3' UTR, so the untranslated region. Why? Because this 3' UTR in flaviviruses is expressed as small subgenomic RNAs, so active RNAs which do not code for a protein but have an activity in themselves. And one of the main activities is to inhibit a ribonuclease, so which is an enzyme, a cellular enzyme, which is able to destroy all free RNA in the cells. So it's a basic antiviral pathway which is inhibited by these subgenomic RNAs. So after a couple of passages, there was a sharp increase in mortality for both strains, and these are what the different mutations are observed. And the only common site where mutations were observed for both strains was the 3' UTR. And we know that this 3' UTR is key for the host adaptation, for instance for dengue virus, but it has been shown for plenty of flaviviruses, and because of this inhibition of the ribonuclease. So what Felipe is currently doing is to see if you take the parental strain of the virus, so the unadapted strain of the virus, if you complement it with the mutated, the adapted subgenomic RNAs, if you can recapitulate and reproduce the high virulence phenotype. This is currently ongoing. Another thing we wanted to know is, okay, we increased the virulence for chicken embryos. What if we compare the parental and adapted strain obtained in chicken embryos, but this time in chicks, 1 week chicks? So we infected these birds, and we compared the response of the PBMCs, so the blood mononuclear cells, and to see how they compare. Okay. For this, we did what we call single-cell RNA sequencing. So you know what RNA sequencing is. You sequence the RNA, the mRNA expression of different cells of the population. But here, it's at the stage of single cells. So first, you separate the different cells, and you can see for each cell how it answers, in this case, the infection. What is interesting is that this is the result from the single paper that there is currently using such techniques in chicken. And you see that there are some leukocytes there, and this is a major population which is not leukocytic at all. And you can see that we observe exactly the same thing in our own data. Actually, these are blastic cells. These are very young chicks. These are blastic cells which are colonizing the different developing tissues. Okay? So we have no interest in these cells actually. But you can see that there are a limited number of cell types and clusters, and we wanted to improve the resolution here. So what we did basically is to bioinformatically remove these plastic cells we have no interest in, and to reanalyze all these cells here. And by doing so, we have a much better, much higher resolution of the definition of different cell types. You can see here all the histiocytic cells with the classical, non-classical monocyte, dendritic cells. Here you have the T cells, so the pre-T cells, and here the CD4, CD8, and the T regulators. And you have here the B cells. There are still some granulocytes which are present here also. So a very fine and detailed, this is the best single-cell RNA analysis there is in birds so far. So this is extremely interesting results. I think this will be a very great paper of Felipe. This is very interesting. Besides, we compared the impact of the infection by the parental strain, so the strain of the virus which is not adapted to chicken, and the one adapted to chicken, and see what changes between those. And you can see that the adapted strain of the virus in monocytes induced a strong induction of interleukin-8, you remember, the one in blackbirds, of different proteins which favor replication of the virus. These are anti-interferon proteins, PAC8 and ATF3, for instance. So there is a promotion of the viral replication, an anti-interferon response, and also a promotion of the apoptotic pathway. This is exactly what we observed previously with the comparison between blackbird and chicken cells. This is extremely interesting because using two different ways, we observe actually that what makes the barrier between different species in terms of resistance susceptibility is made by the same key elements. This was for the PhD of Philippe. What about in vivo? we can't work with blackbirds. I told you it's not allowed in Europe. So we decided to validate a canary model. Canary is a domestic bird, so we can make a model out of it. Then we evaluated the impact of a direct route of infection. I will explain you why. And then we decided to compare the pathogenesis in canaries and chicken. So first, and again, Felipe and Emna participated that. It's published already. We validate this canary model of infection, which is a very good model, actually. Canaries are highly susceptible to the infection. It's a very convenient model of infection. But what we found that was extremely interesting is that there was a shedding of the virus at the level of the skin and the feces of the infected canaries. Also in the literature, we found that there were cases of transmission of West Nile virus in crows during the winter. And during the winter, there is no activity of the vectors. Okay? The mosquito vectors are inactive at that time. So it suggests a non-vector transmission of the virus. Similarly, some colleagues had shown that it was possible to infect birds through oronasal inoculation. So without any injection, without any inoculation, either by a needle or by a mosquito, to birds, to different bird species. We wondered if it was the same for our Usutu virus. Another clue that was interesting in that paper that we wrote a couple of years ago is that in some birds you can see strong viral titers. Here, these are the RNA copies of the virus in the lungs. So there is a strong replication, very similar to the liver and even higher to the brain. So that's interesting. Again, it's a mosquito-transmitted virus. So the question is there a potential direct route of transmission without involvement of a mosquito vector? And this could explain the overwintering, because this virus has become endemic in our regions, in Europe, and during the winter time, there is no or limited activity of the vector, and still the virus stays there, which suggests strongly that there might be a direct horizontal transmission between birds that maintains the virus population. So we compared the intradermal and intranasal routes, and we showed that it was possible indeed to infect these birds, albeit less efficiently. You can see here the seroconversion rate, which is lower in intranasally infected birds compared to which all seroconverted to the inoculation. But still, it's possible to infect birds that way. This is a very interesting image. This is a piece of skin of an intradermally inoculated bird, and this is the nasal turbinates of an intranasally infected bird. And you can see that the inflammatory exudate which is present is highly reminiscent. So it means that the host response is not dependent on the inoculation site. It's really the same. And probably these cells, as we said previously, highly participate then to the spread of the virus to different tissue, like the spleen here, for instance, mostly here in histiocytic cells. First, we wanted to know if there was a shedding of infectious viral particles in the oronasal area of birds. So we infected canaries, we sampled them with swab, and we isolated the virus, and we showed that indeed there was significant amount of infectious Usutu virus there. We also showed that we could predict the possibility to isolate the virus by qPCR. You can see here that high CT values are associated with an ability to isolate infectious virus, while for low CT values, we could, in all these cases, when the CT was lower than 30, isolate the virus in cell culture. We also observed, it's not surprising, it was expected an inverse correlation between the CT value and the survival. So, of course, the birds shedding a lot of virus would die from the infection. We had a dose and effect response. And then we tried to see, because this had been shown for West Nile, if it was possible to experimentally reproduce an horizontal transmission of the virus. So we infected some canaries, and we put them into contact with sentinels in the same cages. And after 15 days, we'd observe if there was transmission, either by the development of clinical signs or by showing a seroconversion of these sentinel birds. Unfortunately, we did not observe neither clinical signs nor the presence of our RNA in these sentinels, nor seroconversion. So it was not efficient. The thing is that there are little contacts between birds, so probably it's not the best situation to try to modelize a transmission. And we thought that in natural conditions, there might be situations which would be more favorable to a transmission. For instance, when birds feed their chicks, chick feeding induces close contacts between the parents and the chicks. And to reproduce that, we use swabs. So we infected canaries, and after 4 days of infection, we just took a swab, an oral swab, and then used a swab to inoculate another bird. And there, it was possible to transmit the virus. So it's possible to observe an horizontal vector-free transmission of Usutu virus under specific conditions in birds, which might be extremely important in terms of epidemiological persistence of the virus, which is called the overwintering, and allowing the virus to stay and to become endemic in Europe as it happens in 2016 now. So this is very, very interesting. Another question we had is, okay, we can infect the birds by the airways, but where does the virus replicate? A French team had shown that the human respiratory epithelium was permissive to flaviviruses, Usutu West Nile. We wanted to know if it was the same in birds. And we did not observe a significant replication, very mild replication of the virus there, using tracheal explants. So we still do not have the answer there. And we really wonder about the cells which are responsible for the initial replication of the virus. So this is ongoing work. Then we compared, and I'm coming to the end of this presentation, we compared the pathogenesis in susceptible canaries and resistant chicken. And this is only starting. You can see that compared to what we observed in mice, here the main replication occurs at the level of the skin, the inoculation site, and then there is a multisystemic spread of the virus, but there is no neurotropism. You can see that here in green is the brain. The brain shows the lowest values in terms of the titers of the virus. This is sharply different from what we observed in mice. So there is no neurotropism there, at least not as clear as we observed in mouse. So it's more a multisystemic disease in birds and more a neurotropic disease in mammals. And this is the work of Ode. We observe a classical, typical splenomegaly that we also observe in natural cases of infection with typical inflammatory response with different leukocytes and so on. And so we really want to compare, because we know that the spread is very limited in chicken. We really want to study in detail what's going on to the skin at the inoculation site of these canaries versus what's happening in chicken, and then the different steps of the spread of the virus in different tissues. So what we are doing at the moment is that there is no validated tool in birds, as you know, and we are a bit like we did for the chicken. We are validating a single-cell RNA sequencing atlas of Serenus carinae, so the canary, because so far there is nothing. There is no information, there is no genome, so still a lot of work. And this is ongoing with Ode. So the first step was to isolate the different cell types. This is for the liver, different hepatocytes and different cell types here. This is the spleen, and to see that these cells are well separated before starting to sequence the RNA of each cell type and have this atlas. And once this atlas will be available, we'll compare the infection of chicken and canaries and see how these cells respond, which are the cell types which are infected by the virus, and how the virus is able to spread starting from the skin infections. That's it for the presentation. I'm open to any questions. Yeah, I suppose there must be a lot of information at the same time, a lot of heavy techniques, quite different from the first presentation, but it's also interesting that we can do some, let's say, high-level technological things also. So the funny part here is that in humans and mice, you have plenty of available tools that you can buy. You have kits to study, for instance, by flow cytometry, the different leukocytes with antibody specifics and validated. You have panels you can buy. It's very easy. You can also use commercial private companies that can do that for you. But for birds, there is nothing. So you have to develop every tool yourself, and that's why these new sequencing techniques are so useful, because it's like being pioneers in something entirely new. Nobody knows what we find. Even the different leukocytes of birds, we do not know them as well as in mammals. I think there was a question somewhere. Yeah.
So I don't know if I misunderstood it, but from the presentation, I noticed that the infection in the days post-infection has like I don't know if the virus has like a tropism for the spleen in birds. Is that like correct or?
Indeed.
Yeah.
We know that there is a strong tropism for the spleen, mostly for the histiocytic cells there in the spleen. When you perform immunohistochemistry to identify the viral antigens, it's mostly in histiocytic cells. The thing is that we do not know specifically which cell type. And we don't know why these birds die from the infection, because the lesions are not so impressive. Okay, the virus is there, there's inflammation, but it's not so impressive. So we strongly believe that there is an inflammatory dysregulation, like the cytokinic storm, a multisystemic failure of different organs, but we still have to study.
Oh, okay, yeah. Even if it doesn't have like a neurotropism, it does have like neurological symptoms, and that's why the
Yes.
die.
Well, the thing is that it's always hard to differentiate between, okay, when you're severely sick from something, it looks like, you know, anorexia and so on. It's hard to tell if it's pure neurological or just multisystemic. It's not easy to make the difference. There are no, I didn't show you the image from the infected mice, but in the mice, it's very pure neurological. There are paraparesis, for instance, or ataxia that you do not observe in birds. They are just sick, and that's all you observe, and then they die. Okay. So also probably it's not exactly reminiscent from what was observed in blackbirds, natural blackbirds. But again, this virus has been passaged a couple of times in the laboratory cells. It's probably exactly the same. But this is another project. I'm not tell you about that today. When you inoculate using a needle a virus, it's not the same as using a mosquito. Because in mosquito saliva, there are different molecules that change the behavior of the virus, but that's another story.
Sorry for asking so much, but like, is there also like a difference with like how the virus develops in, for example, blackbirds and raptor birds?
We do not know, because it was hard to do in blackbirds, because getting cadavers was complex. It would be even more complex in raptors. Actually, we had isolated such cells from raptors also, but we did not do this analysis because it's a lot of work. Could be done, yeah, but we didn't.
¿Se puede preguntar en español?
Sí, por favor.
¿Hay alguna proyección epidemiológica acerca de cómo pudiese dispersarse las distintas rutas migratorias, especialmente pensando en nosotros como América, en estas rutas polares? ¿Hay alguna especie que se haya visto significativamente afectada, que tenga esa migración?
So I'm not sure I got the whole question. So the question is about the prediction about the spread of the virus through the migratory flyways, if I got it correctly. So yeah, certainly. So a bit like, I think the avian influenza is very interesting on that respect, because it emerged in China. Then through the flyways, I could show the flyways again, but it was able to reach Europe, and then the Americas. So it became global in a matter of a few years, very easily. Okay, so there is no reason why it would not be the same for other viruses like the flavivirus. We know that West Nile did that a couple of years ago in North America, but it didn't reach South America. We do not know why. But it might happen anytime soon. So let's see how it evolves. That's also why I think it's important to have tools to be able to implement surveillance program quickly if something is happening. That's why in Europe there are heavy efforts to sample migratory birds. In the Netherlands, for instance, the Erasmus Medical Center is sampling a lot of wild birds on the seashore to see what's circulating there. It helps. Yeah?
I see some virus, avian virus, to have resistance in domestic animals, like ducks and chickens. Since in raptors or other passeriforms, they die in few days. Maybe that have the answer in the artificial selection by humans in that types of birds, or is maybe a genetic answer?
That's a very good question. No, it's not related to domestication because in wild anseriforms they are resistant as well. So they are wild birds. So it's really related to the order of the birds. And it evolved over time. So in the first times of when the virus was really arriving on the continent, it killed a lot of birds. But then there was a group immunity appearing, and now the resistance of these birds has increased because there is an equilibrium between both. A bit like we observe with SARS-CoV-2 in people, for instance. So there's a new trend. But still, we are observing a lot of mortalities. We had a large number of mortalities in sparrows also, with a sharp decline in the populations, and now it's starting to increase again. So it's not related to domestication, no, I don't think so. Usually it's the opposite, because the genetic diversity decreases with domestication, and usually things are worse. Any other question?
Preguntas, comentarios, Espero que los estudiantes de fisiopatología hayan entendido todo. Comentarios, yo creo que bueno, además de agradecer al profesor, nos ha mostrado toda su experiencia, su trabajo en el diagnóstico y en la vigilancia epidemiológica, complementando el trabajo de sus entidades gubernamentales, así como ocurre también en nuestro país, nuestro trabajo con el Servicio Agrícola Ganadero histórico a través de los años. Todos se dieron cuenta que la charla tiene epidemiología, biología, que trabaja ahí. Hay algo muy interesante y destacable que es, no basta con diagnosticar una enfermedad, sino que hay que crear modelos para poder estudiar. Y eso es algo muy complejo, ¿verdad? Ustedes pudieron ver ahí resultados positivos, negativos, vieron a nuestro amigo Felipe también, pero la creación de los modelos de patogenia creo que es algo tremendamente destacable del equipo del profesor, y por supuesto los comentarios que hace de la importancia de la medicina veterinaria en la salud pública. Todo eso lo tenemos bastante claro. ¿Hay alguna pregunta más? ¿Comentarios? Le damos un fuerte aplauso.
I think there was a question there.
You showed some species. I don't remember if they were rats. Some were like 9 days old.
For the mice, you mean?
Yes,
Yeah.
the mice. Yeah. I didn't get the reason, because why was a much higher mortality on the younger ones?
Yes. So this is, as I showed you, it's something which is well known for flaviviruses, but it's well known for many viruses. There are several possible explanations for that. The first is that there is a Th2 bias. So you know that the adaptive immune response, you know that the Th1, Th2, well, there are different orientations, I would say, of the adaptive immune response. And among the Th2 response are notably allergic disease, which are more frequent in young individuals. And an efficient control of infections usually require a strong Th1 response. And this Th2 bias might be part of the explanation of the higher susceptibility of young individuals to the infection. But the thing is that if you see the kinetics, the difference occurs so early in the process that probably it's not due to the adaptive immune response. Another thing is that the central nervous system in young mice is not mature. It's still developing, and there are still a lot of incoming cells. I showed you in the blood of this chicken, for instance, that there are plenty of blastic cells, which are produced in the bone marrow, for instance, or in different tissue, and then circulating into the blood, and then gaining access to different tissues. And this is the same for neuroblastic cells, which are still arriving, including macroglial cells, so which are monocytes deriving into future macrophages, local macrophages. And this could be, we call that the Trojan horse hypothesis. They could carry the virus into the central nervous system more efficiently in young mice because the blood-brain barrier is still permeable to them, and not in older mice. That's why we want really to focus on what's going on with the spatial transcriptomic approach, seeing what's going on there and how the virus is able to cross it. So maybe just one comment after what Thomas Casanova said. If there is one take-home message here, is that very classical old virological methods like viral isolation, the development of in vitro tools, basic in vivo models, cell neutralization can do miracles, are really important. And we realize with the SARS-CoV-2 episode that not so many people can do that anymore. Because we use brand new techniques like I show you single cell RNA sequencing, okay, it's fine. But when we ask, okay, who can isolate the virus? Not so many people can do that anymore. And that's what makes the difference. That and having BSL-2 and BSL-3 facilities. We had a discussion with the rector lately, and to me, this is a priority. If I can give you a piece of advice, have that kind of facility and people that can do that kind of work, it makes really the difference. That's it.
Hacer un comentario por acá de preguntar, pero más que preguntar puede ser este comentario ustedes vieron a nuestro exalumno Felipe Rivas en varias de las fotografías. Es simplemente un mensaje de motivación, de creer que se puede seguir estudiando, que se puede viajar a otros lugares del mundo a conocer esta tecnología súper avanzada, ser parte de tremendos equipos de trabajo. Simplemente hay que querer, esforzarse, el apoyo nuestro lo van a tener para partir a diferentes lugares del mundo. Uno de ellos podría ser la Universidad de Lieja, ¿verdad?
¿Por qué no?
Claro. Eso creo que es un mensaje para nuestros estudiantes. Vuelvo a preguntar si es que hay alguna pregunta, algún comentario más. Bueno, también un saludo a los que nos están viendo online. Y ahora sí, entonces le damos un fuerte aplauso a